So our next speaker is Dr. Adelaide Rhodes. So Dr. Rhodes has more than 20 years of experience in analyzing big data at the molecular, organismal, and ecological scale. She holds a PhD in zoology for NCSU and an MS in biotechnology and bioinformatics from Johns Hopkins. As a bioinformatician and strategic consultant, she has assisted dozens of universities and government researchers to analyze cancer, crop science, bacteria, virus and plant genomes, transcriptomes, and disease variant datasets. She has developed advanced workflows for secondary and tertiary analysis and has written custom scripts in Bash, R, MATLAB, and SAS to interrogate large datasets for meaningful signals. She also has a strong publication record that spans multiple disciplines. Recently, she has helped researchers develop pipelines for data-driven discovery, such as HAIL, GWAS, and CNN, using a variety of workflow languages like WDL, CWL, and SnakeMake and Nextflow on HPC and commercial cloud computing resources like AWS and GCP. She also provides consultation on AI and ML genomic opportunities for data-driven discovery in the health and life sciences. So now I would like to turn it over to Dr. Adelaide Rhodes.
Hi there. Should I share my screen for my PowerPoint?
Yes, please. Are you able to do so?
Yeah, let me share screen and pull it up. I wasn't sure about the meeting format, so let me do that. Oh, great. Can you guys see my slide?
Yes, we can. Thank you.
Okay, good. This is going to be a little different. And thank you, Alice, for that presentation. I'll just say that I use Google Cloud. You might hear a little bit of that when I talk about what I've been doing. And yeah, so what I'm going to talk about today is, wow, that sounded really impressive when Laura read out all those like qualifications and what I do, but I didn't just start out that way. So my talk today is a little bit of a motivational speech about anybody can get to where I am if you just put your time and effort into finding ways to kind of get involved and improve your skill sets. So I do have to say that because I work for an NGO now, like the views and opinions are only my views and opinions, and they're not a representation of any organization to which I currently belong or I have previously belonged. Okay, so I'm gonna walk you guys through my little bit of my life story and then give you at the end talk about lessons learned. So in order to kind of give you guys a bigger picture, I'm just gonna talk about being a scientist and then switching from being a scientist to becoming a bioinformatics faculty member at a university, and then realizing that wasn't a fit, and then moving on to working in bioinformatics full-time, mainly with cloud content. And then, of course, we all kind of experienced the pandemic. So what does that do to our careers? How does that look like? And then is this an opportunity, not something that hurt us? Okay, so just to give you an idea of where I was 20 or so years ago, I studied copepod crustaceans, which are at the bottom of the food chain in the oceans. They're very important species for fisheries. There's been 75,000 described species, and I spent most of my time looking through a microscope at these organisms and trying to describe them. And then I later got into DNA barcoding. That was my gateway into bioinformatics because I was generating a lot of sequences for unique organisms that were being collected here. You can see Sherry Reed and another, oh, that's me down below in the lower right corner. I used to go out and collect benthic species, sort them, look at them under a microscope, but it would be really nice to know if this had ever been observed somewhere before. So this was near the beginning of the Barcode of Life project. So I contributed a lot of marine species to that effort. Basically 18S or COI, cytochrome oxidase 1, species sequences. So this was actually back in the day, like we're running them in the lab, had a Sanger sequencer to put them together. And the frustrating thing was like, I would go onto the databases and I'd be like, and no one's ever seen this before. and that pretty much happened for most of the species I was working on. I was cataloging them to be of service, but a lot of them were unique observations. And then this happened. So I was working in Florida around the time of the Deepwater Horizon oil spill. And what's happening here is that we had to collect a lot of data really quickly. So this kind of pushed the boundaries of what was being done in our lab to include a lot more thought about how to collect a lot of data about a lot of different species over several years. And I was put in charge of not the copepod crustaceans, but the Tenea crustacean observations. And you can see here on a slide, I had to look back in time. I wanted to know what had people observed previously so I could understand what I was observing now. Once again, just validating whether there were new species in this region or not. So it did involve data, but it didn't involve any genetic data. But I did want to do genomics because we were working in a very interesting and unique environment, the deep sea. So deep sea environments are really different. Not sure how much you guys know about it, but there are coral species at the bottom of the ocean that do not require photosynthetic organisms, the zooxanthellae, to live. There's a lot of symbiotic organisms, things that, because it's very sparse, there can be a large distance between species or little species islands. Things have learned how to grow up together. So there's just a few pictures here of the pretty corals and the commensal species, the brittle stars that hang on the corals, but there's so many examples of that. So it's a really unknown environment. And so getting a really good list of species that were at the different sites was really important. And as you can imagine, being very good at organizing the data, being able to access the data, tens of thousands of observations, plus observations on the conditions, the biochemical conditions at the bottom of the ocean were important too, because we were kind of trying to build data layers, like looking at the species, looking at the chemistry, looking at physical conditions, looking at where the plume was flowing. That all was very complicated and resulted in quite a few papers. So what I realized through this process, and where am I going with this, is that I really like the data part. And so I did get a chance to do some more barcoding type genomics as a side project during this time. So I really realized two things about myself, that the parts I liked about my job, believe it or not, as a marine biologist, did not involve going out to sea. They actually involved the part where we had a bunch of data and a lot of spreadsheets, and then we had to tell a story. We had to say, this is what happened here. This is what we need to pay attention to. So looking for patterns, trying to come up with hypotheses and then testing the hypothesis all on a computer. It's fascinating stuff. So that's when I decided to make a career change. And it didn't happen overnight. I did go back to school, even though I had a PhD. I chose the Johns Hopkins University program in biotech with a major in bioinformatics because, well, first of all, it was one of the only two that were available online, 100% online at that time. The other one was Stanford, and that Stanford was double the price. And the other thing was for the amount of time you had to invest, it was very comprehensive. Like you took molecular biology, you took computer science classes and all that. So you have to understand my PhD was in biology, didn't mean that I knew how to do bioinformatics. So I really, really appreciated that program. And so as a result of getting a master's, I actually was able to get a professor job over at Oregon State University. So that's what the little beaver guy is there. And so I ended up moving over to Oregon State, and the best way to learn is by teaching. So even though I was a very new bioinformaticist, I taught classes before. So I thought about, what do I want to learn? And then I started teaching things I really wanted to learn about. And I taught them several times a year to graduate students, undergrads, and then other people like myself, basically a lot of people who came up through school, finished school before any of this existed. So I had to, construct classes. I'd had to understand how to do the data analysis. So it was a great way to learn how to do more more. And so that's when I also realized what is my favorite programming language. It's Bash. So I really do like Bash, and it's just the cleanest and stuff. But I did have to know R and Python. So being in a school, teaching, volunteer teaching, mentoring, for those of you out there who feel a little bit of the imposter syndrome, it really helps you get past that, because you're transmitting, you may only be a couple steps ahead of somebody, but you are a couple steps ahead of somebody. So by transmitting that information and getting involved in projects, I was able to kind of overcome that. And then the other thing that I got to do, which was really fun, and if you notice at the beginning, the little guy holding up the sign is a beaver. That was the mascot of the school. So I was actually put in charge of a really cool opportunity to teach students through generating hands-on experience with a genome assembly project. And of course, we picked the beaver to do the genome assembly, which meant that the students and other professors that were working on the project, we were all involved from the very beginning to the very end. Data collection, data analysis. What did we learn from having another rodent genome? Because of course, mice and rats are fundamental organisms for medical research, but there isn't a lot of diversity out there at that time for other genomes that you could look at for other answers. And so there's a few features of the beaver that aren't present in other rodents, such as the castor sac. They make a castor oil, and the fact that they live most of their lives in dens in the water, so they do have to be able to hold their breath a very long time. And another feature about beavers is that they are ecosystem engineers. So their brains may develop differently. So I was in charge mainly with the transcriptomic part, and then cataloging. We didn't really find any new genes, right? We just found homologs, but there were long non-coding RNAs that were very interesting that might have shown how beavers evolved over time to take advantage of the environment they lived in. And so that resulted in this paper about pan-tissue transcriptomic atlas methods to find long non-coding RNA. So this was the first paper where we actually built a new pipeline, something that hadn't been done before, in order to quickly screen for long non-coding RNAs in non-model organisms. So just think about the fact that, like, if you haven't had a lot of chances to get involved or learn, this is an opportunity, like maybe on your own campuses, at your companies, like there's always a way to get more experience. So volunteering or finding opportunities to help other people do their projects is a great way. My job was in the core facility. For those of you who don't know what a core facility is, it's basically a service that is provided by the university. So anybody studying anything can come into your office and ask for help, and the university subsidizes the salaries and the supplies for people in the facility. So I'm just going to flip through a few different examples. And so these were not research projects I initiated. These were ones where people came into our facility, asked for help, and it was my job to try to get them the best answers I could. So we looked at viruses and omycetes, which are slime molds. Another project I got involved in with Molly Burke's lab was a molecular yeast evolution pipeline upgrade. So that was an automation project. I also was co-author on a paper about canine human bladder cancer comparisons. That was very interesting because we not only got to do a deep dive into canine cancer, we actually looked at human cancer and combined data sets. So that was really comprehensive study that was very challenging. So I learned a lot from that, and I also got into a little bit of the modeling. And another one was looking at pathogen-associated molecular patterns, so basically innate immunity in bugs. So I did more than one of those. Stink bug was a more well publicized paper that I was on, but there were, like, as you can see, there's like couple dozen people on the paper. So you can get involved, you can publish. You don't always have to be the person initiating the idea. Like if you're willing to pitch in and look for unique patterns, or you're asked to because it's your job, you can certainly do research. And then I did continue my own research, looking at copepods. And so I was using reduced genome methods, to be rad, to try to find biomarkers at a time when, on the right of the screen here, you'll see like a little map with pins in it. That's the coast of Oregon and California. And what's happening is there's a warm water front that appears every few years called the blob. They really do call it the blob. And so the idea there was, well, if there's this large patch of warmer water, could copepods from southern regions be migrating into Oregon and sticking around? Now, who cares? Like, right? But copepods are the main food of salmon. So if they're not recognizing the species of copepods that have come into the area because of the warm water, are they going to have problems maintaining their body weight and living through their maturity? Because salmon come from rivers in Oregon and California, and they go out to the ocean and they live in the ocean for a few years. And then they need to have all this energy stored up so they can make it back into the rivers in order to, be able to spawn and pass on their genetics to other salmon. So it was a really interesting project. Still went out to sea a little bit and generate some novel data that kind of showed that, yes, there were some stratifications in the populations that hadn't been seen ever, before. And that was enabled by genetics, genomics, and big data. Oh, and I just throw this picture in here because we were also having the pyrosomes. There were like a million things called sea pickles. So we'd throw out our nets and we'd pull them in. And instead of getting a bunch of fish and flounder and stuff, just FYI, we got these things called sea pickles, and they were a big deal that year. And that had a lot to do with global climate change, right? Because this warm water area, other species were coming in and taking advantage of it. So I could have been happily content to work on other people's projects and everything, but I had also been getting really fascinated with cloud computing and using cloud to answer questions. So this was about 7 years ago. I mean, now we all use cloud, and cloud is accessible. We just had a presentation talking about cloud, but I really, really wanted to go in that direction with my career for some reason. So I ended up moving to Boston, and my first job actually did involve Google Cloud platform called Terra. If you're not familiar, it's at the Broad Institute. And the idea of a Terra is that it brings together all the resources you need to reproduce a study. So you may know the pain of reading a paper, a very interesting paper, and you're like, oh man, I could totally use that idea. And then you start reading through the methods and it says, produced by custom scripts, that are, and then you'll call the lab and they'll be like, oh yeah, those custom Perl scripts, disappeared when our grad student left the lab. We have no idea how they actually did the analysis, right? Or there's a data set, there's like a spreadsheet, and, it's maybe just one of the pieces of metadata that was used to generate the final product, but it's a very important part, but that's not in the supplementary materials. You can't find it again. And then you call the lab and they're like, we don't know, that was like on a book drive, and then we had a fire and then we don't have that anymore, right? That happens all the time. That's why cloud is so important, because it gives you a place to put all your stuff. And Terra is cool because you can put together your data sets, you can put together your methods, and then you can actually run really high throughput workflows using things like the workflow development language, or CWL. So you can use your free credits. Like I heard the person before me, Alice, just talking about that. You can use your free credits to play around with Terra. It's mainly biomedical research, but there's a lot of really cool, interesting stuff on there, and you can read it for free. Like you don't actually even have to sign in to get a lot of value out of it, because you can flip through the pages and see how other people did the program, or how someone wrote their WDL, or you can find Dockers. Like you might need a Docker for Tidyverse or something, right? Like people have written that like over and over again. So that kind of stuff's available in Terra. Thank you for putting the link there, Laura. It's terra.bio. But anyway, I don't work there anymore. I really did enjoy my time there. But I really wanted to not be stuck in one type of role. As a scientist, I kind of wanted to explore and do more diverse things. But I'm also just putting this up here. This was one of the products out of my work at Terra was like a synthetic BAM generator. But you can see here this arrow. If you are not familiar with the FAIR principles of data science, I really feel like it's worth reading up about because there's lots of opportunities. So once again, if you're starting out and you're not quite clear which direction to head in your career, but you do want to make a contribution, FAIR is a way to kind of get involved and know like how to present yourself, how to present your data, how to make things reproducible. Because if people can find your stuff and use it very easily and reproduce your research, they're more likely to cite you. So if you're still on a science track, just thinking a little bit about a project before you start it is going to help you. And so I just gave a talk to my coworkers today about being FAIR. So it just means findable, accessible, interoperable, reusable. All right. Don't make a spreadsheet and then don't put it in supplementary in your papers, in other words. And then but data was getting bigger, and like I said, I wanted to get more challenge out of my work, so I went to a third-party partner for Google Cloud. So I actually trained people. So I wanted to get beyond just doing a thing, and so once again, I wanted to learn how to do it better. And for me, my philosophy is if I teach somebody how to do it, I can learn how to do it better myself. So I'm very fascinated. This happens to be a Google schematic, but there's AWS corollaries. There's also, Azure. I'm not trying to influence any kind of product decisions, but basically you can build up from, I know how to use a spreadsheet, to, okay, so I can take that spreadsheet and put it into a database. So now I know how to use BigQuery, and I know BigQuery, oh, wow, that's going to enable me to ask interesting research questions through machine learning. And then, well, maybe I need a model for this thing I'm trying to do. I'll go over to the Google Brain and look for some advanced protocols, right? So I like the versatility and the flow of Google, so I tend to default there. But like I said, there's really great choices on all the cloud providers. And this is just an example of kind of a product that I came out with, showing the power of cloud, because we are generating genomes at a faster rate than anybody ever anticipated. And even just knowing where to put them is a challenge. So cloud, it's all about like, how do I store a large amount of data? How do I access that data? And then what can I say about the data after, everybody else has done all this really important work, putting the data where we can get to it? What can I do with it? So those are the kinds of questions you should ask yourself as you're going through your career. Like what parts do you like? Where do you want to be? And then like I really got into Kubernetes and Nextflow and all that through those experiences. And one thing I found I lacked in, and I went back and got some more training through Coursera, et cetera, was how to present the data back to the researcher. So I spent a lot of time thinking about these interactive tools such as our Shiny or Plotly or Observable, right? There's a million of them popping up now because that is a common need. You could be somebody who specializes in that. Like if you particularly don't like any of the database stuff, or you don't particularly care about the infrastructure, just knowing how to communicate is so valid. And these types of things are very easy to learn. And there's tons of free documentation out there, Coursera, Udemy, GitHub itself. Like people put up very nice and web pages and share like how to build these like visualization and interactive tools. And then that resulted in me like doing some work where you can find these videos out there on the internet if you care about, but like trying to fill in those gaps. Like I'd be like, man, nobody knows how like the NCBI is on the cloud, right? Like everyone keeps hearing that NCBI is on the cloud, but like, how do you actually like go to the cloud, get the data and use it for something? That's the gap. That's the gap that I keep trying to fill. That like, yes, it's on the cloud. Cloud is great, but if you can't have a concept of even what that means, you're not even going to be able to take advantage of all these cool, freely available free resources. Because even though Google and Amazon and Azure cost money, they all have free tiers. And I heard a little discussion about credits. There's ways to get credits. Like when you sign up for Google, you get like $300 right away. There's ways to kind of dip your toe in the water without spending any money at all, but getting the benefit of thousands of researchers' data. And so I've seen this a lot with the pandemic data, like a lot of that was made publicly available. And there was a kid here, like his high school project was making a dashboard for the data that just kind of updated people, and it got a lot of buzz. It was really popular. And then now he just went out and made a dashboard for Ukrainian refugees, like people who want to share their houses, connecting them, because this data exists. There's just a gap in, like, how do I get to it? How do I find it? How do I use it? So that's a really good opportunity. And then, as I mentioned, there was a pandemic. I didn't build a dashboard, but I did other things. I spent a lot of time like on trying to develop skill sets through hackathons, and by volunteering once again to teach through the Bioinformatics Virtual Coordination Network, which has a bunch of free lessons like on RNA-seq, metagenomics, genome assembly, gene annotation, things like that. All right. This is my last slide. So just a little thought about the different categories of bioinformatics. As you can see, it's a really diverse world. Not everyone's going to be like me, where they have a short attention span and you're going to jump from one to the other, right? But that's my style. That's who I am. So I would say to you guys, be who you are, figure out which part you like, invest time in yourself, always be building your skill sets. Like I said, you don't have to spend a lot of money. There's so many free things out there. Networking, this group is great. There's always so many cool opportunities on the LinkedIn coming out of the group that is sponsoring the symposium. Reaching out to people like a little further ahead, like 2 steps ahead of you, because like I said, I'm never like put off by somebody asking me for help, because I'm always like, oh, that's a great opportunity for me to learn this thing better, because now I'm going to have to explain it to somebody how you do this thing, right? So think of it that way if you're shy or you're afraid of asking for help on a particular project. But yeah, that's about it. That's all I had to say. This is me. This is who I am. And I guess I have a few minutes to get some questions. And I didn't, okay, here we go. Let's see. I did see that Sona said that you do diversity and ecology jellyfish. So hi, Sona. Yes. And then
actually, it's not me. I'm sorry. It was I posted it on behalf of Gisele.
Thank you.
Because I think he didn't have or.
Thank you. Thank you very much for posting that.
And yeah, but I must say, now that I'm online, I know this is out of my turn, but extremely inspiring talk, amazing talk. Thank you.
Thanks. Yeah. And then there's one here, question 1. How do you compare working for academia versus working for the industry? That's a great question. I'm an impatient person. So sometimes for me in academia, things didn't move fast enough. But I also don't have all the energy I used to when I was younger. So the problem with industry, sometimes it moves a little too fast. So I'm looking for the Goldilocks job all the time, where it's not too slow, but it's not too fast. In academia, the resources are limited, but that means your creativity gets an opportunity to do the work. That's why I know about so many free things, because as an academic, I had very little money. So, I had to accomplish big ambitions with nothing. So I hope that answers that question. Question 2, how do you decide what kinds of research projects to focus on to advance your career? That's a great question too. And because I'm the type of person who says, do what you love, I also like to love what I do. So my career decisions a lot of times have been led by, I just don't love this anymore. This is not something I see myself doing like for 20 years. And so then I'll just go to the next level. But that's not very practical for those of you who, may not have the ability to move around like I can. So I would just say, look for opportunities in your company. There's always somebody who needs like a hand or help. And you never know, just like by volunteering a little, spending that extra hour with a project per week, showing up for a meeting that you weren't necessarily invited to, but you heard about and got permission to attend. Those are ways to kind of edge yourself over. You don't have to make leaps like I do. You can just edge yourself over. And then, of course, taking the trainings online are very helpful, because then you show up and you're now the expert, like maybe in your group, because you spent a little bit of extra time on Coursera or in the Pine Bio, platform, learning how to do a workflow. And it's needed all the time. Like, other people just don't have the time to look. But if you have a little bit of time to look, invest in yourself. I only got 2 minutes left, but question 3, how has bioinformatics changed over the past decade? It's just gotten bigger and harder in the fact of, storing data, but it's gotten more interesting and impactful because you don't have to collect the data to be the person to say something about the data. So that's pretty cool. Let's see. Sorry, I'm going to skip it. Manor says, I'm a student of biochemistry. I love to learn more and more about bioinformatics and genomics. Yes, people, you please feel free to reach out to me through LinkedIn. I'm not sharing my email because I don't check my email enough to be a great person, but LinkedIn, I see it pretty regularly. So do that. And then last question from Elias is, what is a major skill students struggle with on their path to mastering bioinformatics? I think it's confidence. I don't think it's even a skill. I think it's just believing that you are. So a lot of these changes I made was me saying, I am that now. I am a bioinformaticist, or I am a data scientist. Nobody waved a magic wand over my head and made me that. Like, so that's why I'm saying that students struggle with that. So just be the thing that you think you are, and you are. So, and then the BVCN is just a freely available set of lessons on GitHub. It's called the Bioinformatic Virtual Coordination Network. I do link to it in my LinkedIn profile. You can see it in the featured pages there. Like, there's links, and there are videos and basic lessons. It's all free. It's all freely available. Just how to do metagenomics. It's mainly metagenomics, but there is some transcriptomics. But for beginner bioinformaticists, it's a nice little set of lessons that we wrote.